dental and genetics. You don't have your usual host, Dr. Kabashi, as she was unavailable to host today. And we are very pleased to have Dr. Samantha Cairncross joining us today to host the webinar. Dr. Samantha Cairncross is a postdoc fellow at the NRF Temba Lab, specializing in radiobiology, radiogenomics, and precision medicine. Her research focuses on identifying genetic biomarkers associated with radiotherapy-induced toxicities in cancer patients, with a particular emphasis on African populations. Okay, and she's also a recent University of Western Cape graduate. Dr. Cairncross, I hand over to you.
Thank you so much, Iman. I'll share my screen now and I'll begin.
Sure.
Okay, good afternoon, ladies and gentlemen. So I am Samantha, and today I'll be presenting a part of my PhD work titled The Investigation of Radiogenomic Biomarkers for Radiotherapy-induced Toxicities in Head and Neck Cancer Patients. So cancer is an emerging health problem in Africa that needs to be addressed in order to control for increased incidence and mortality rates. And so even though cancer deaths have surpassed those of AIDS, tuberculosis, and malaria combined, there remains a lack of commitment to fighting cancer in Africa. And it has been suggested that by 2030, there will be a 70% increase in new cancer patients due to population growth and aging. And cancer mortality rates are expanding in Africa. And radiotherapy will increasingly play a critical role in improving disease outcomes. Okay, now my screen is freezing, I'm sorry. Is it moving on the other side? I'm sorry. So radiotherapy is an important part of the cancer treatment regime for the cure and/or management of various cancers, with nearly 50% of cancer patients undergoing radiotherapy at some point in their illness.
So,
and in
Samantha,
low, sorry, yes.
are you sharing your slides? Stop sharing.
Did it stop sharing? Oh, sorry about that. It froze from my side. Should I just start over again?
Yeah, you can maybe just reshare them.
Okay, just share quickly. Okay. Can everyone see my slides now?
Yes, we can. Can you maybe move to the next slide?
The next one? Okay.
Oh, yeah, okay, it's moving, yeah.
Okay, perfect. So radiotherapy is a very important part of the cancer treatment regime for the cure and or management of various cancers, with nearly 50% of cancer patients undergoing radiotherapy at some point in their illness. And in low to middle income countries, this can be as high as 70%. This is because it's considered a quite cost-effective treatment, and most patients represent with a very late stage of their cancer to the clinic, in which a multimodality treatment plan is required. Okay. So radiotherapy uses high energy X-rays to target and destroy cancer cells. And especially in the case of head and neck cancer, more than 75% of patients will benefit from radiotherapy. So radiotherapy can kill by either directly interacting with DNA, and that can lead to cell death, or it can indirectly interact with water molecules, resulting in reactive oxygen species and These reactive oxygen species in turn damages the DNA, which can result in cell death. So we can see over here that radiotherapy is given in fractions. So when we give radiotherapy to cancer cells, we can see that during these fractions, they are unable to recover from the radiation that they have received, and ultimately the damage accumulates, and that results in cell death. Whereas in normal cells, between fractions, it allows cells to repair, and that is how radiotherapy selectively kills cancer cells as compared to normal cells. So despite significant technological advances in radiation delivery systems to enable more precise targeting of the tumor cells, the unavoidable exposure to normal tissues to radiation can result in significant side effects, remaining a critical concern in clinical practice. And toxicities are especially important in head and neck cancer cases, as in the field of radiation treatment is very close to critical structures, so it can compromise essential functions such as breathing, eating, speech, as well as have any aesthetic concerns. So we can see that to selectively kill cancer cells, we rely on normal cells being able to repair themselves really quickly. But in some cases, or in some people, there's normal cells, either because of differences in DNA repair mechanisms or DNA damage responses, cell cycle regulation, or apoptosis, or any type of defense systems, then normal cells are not able to recover, meaning that when the radiation gets exposed to the normal tissue around the tumor, this normal tissue can become damaged, and that results in side effects for the patient. And radiotherapy kills mainly as a factor of dose, where an increased dose increases the probability for cure, but normal tissue sensitivity limits the dose that can be given. And normal dose constraints in clinical practice are generally determined by a therapy dose of a 5% probability of serious toxicity within 5 years. So we can see that with radiotherapy, if we increase the dose, we increase the chances of cure. But as soon as we increase the dose, we are now having more and more patients being exposed to severe complications. And these complications or toxicities can either occur early or late. So early toxicities can be defined as toxicities or side effects that occur during treatment or less than 3 months following treatment, whereas late toxicities have an onset of more than 3 months after treatment. So in the case of head and neck cancer, when we look at early toxicities, we can think of things such as dysphagia, mucositis, dermatitis, as well as xerostomia. And late toxicities, we're looking at things like osteonecrosis, dysphagia, trismus, shoulder weakness, as well as radiation ulcers. So early toxicities can cause the hospitalization of a patient. It can increase the cost to constantly treat these toxicities. And when we look at the context of South Africa, our medical care is out of pocket. So it increased the costs not only for the patient, but for the hospital, etc., etc. It can result in treatment delays for the patient or negatively impact compliance, meaning that the patient has not enjoyed the treatment because of the side effects and will not be compliant or stick to the schedule. And both treatment delays as well as this negative compliance can worsen the prognosis for the patient. Late toxicities, on the other hand, they are irreversible. They can worsen over the years. And when they come back, for example, the radiation ulcer, they can mimic things like recurrence. So this increases survivor fear as well as anxiety, and in general can just worsen the quality of a patient's life. So toxicities can place a huge burden and stress on individuals, on families, on health services, as well as on society. But the onset of toxicity show a large patient-to-patient variability in both the incidence and both the severity. So despite patients having similar treatment protocols, for example, radiation dose distribution, or any concurrent chemotherapy, and there's evidence that radiosensitivity is intrinsic to the patient, meaning that it can be caused by genetic factors. And as a matter of fact, research suggests that genetic differences account for approximately 80% of the interpatient variation that is observed. And because it is genetic, these side effects remains unpredictable. That means that the oncologist cannot look at a patient and based on their genetics really predict who will experience side effects and who will not experience side effects. So single nucleotide polymorphisms have emerged as a potential biomarker or a potential genetic biomarker for predicting radiotherapy outcomes and adverse effects. So single nucleotide polymorphism is simply a change in one of the nucleotides, so it's a one nucleotide different change, but it can affect gene expression as well as any protein functionality. And these genetic variations can influence the DNA repair mechanisms, it can influence cell cycle regulation and inflammatory response or any other responses, all of which are critical in determining the tissue response to radiation exposure. So personalized medicine takes into account the genotypic, phenotypic, demographic, and clinical information of an individual patient to provide a more personalized treatment response. And pharmacogenomics is a field, a subfield of personalized medicine in which they focus on tailoring treatment based on a patient's genetics. So currently, most treatments or most treatment regimes involves giving a patient the same type of treatment schedule or same type of drug regardless of differences. And this results in only some patients benefiting from the treatment, some patients having no benefit from the treatment, and others experiencing adverse effects to the treatment. But with pharmacogenomics, there is a hope that based on a patient's genetics, that we can tailor a patient to predict these responses, and we can tailor the type of medication to the patient. And so while pharmacogenomics has made significant strides in personalizing drug treatments, similar principles are increasingly being applied to radiation treatment, and this field is known as radiogenomics, where we try to tailor a patient's treatment based on their genetics. So what are the advantages of actually using genetics when it comes to radiotherapy treatment? Well, we can stratify patients. And so we can see that where there's patients that are more radiotolerant, they can have their radiation dose increase, and therefore we have greater tumor control. Because as you can see, if we can allow for a greater increase in the radiation dose, we now increase the chance that there will be a cure, or that it can be cured. And when a patient is more radiosensitive, we can either, have different treatment options being presented to the patients, or have them have alternative radiation schedules. So over the past decades, candidate gene association studies as well as genome-wide association studies have discovered numerous potential genetic predictors of radiosensitivity. And the field has significantly grown in recent years with more candidate gene approach studies, more genome-wide association studies, more and meta-analysis, and other prospective validation studies. For example, Cairns et al. Conducted a meta-analysis with 6 genome genome-wide association studies in men of European ancestry who underwent radiotherapy and identified RS17055, 178 with rectal bleeding, and RS109699113 with decreased urinary stream, and RS1112253 with hematuria. However, the majority of these radiogenomic studies lack ethnic diversity and many populations are not included. A systematic review by Zuber and Yaya in 2021 revealed that existing studies are mostly homogeneous in terms of race or ancestry of included patients, with a notable absence of studies on sub-Saharan African population. And additionally, to our knowledge, till this day, no radiogenomic studies have been conducted in the sub-Saharan Africa population presently. So the deficiency in understanding specific genes crucial for treatment personalization in developing nations will further exacerbate existing disparities in cancer treatment outcomes. So the aim of our study was to investigate the genetic basis of radiation-induced toxicities in head and neck cancer patients by identifying associations between SNPs and early and late radiation-induced adverse effects, ultimately contributing to the development of personalized radiotherapy approaches. So we obtained ethics approval from the Human Research Ethics Committee at UCT, as well as the Biomedical Research Ethics Committee at UWC, and we got special permission from Groote Hospital to conduct the study. Furthermore, we recruited 162 head and neck cancer patients from the hospital, and inclusion criteria included that they had to be older than 18 years old, they should have given informed consent, and they should have had a confirmed lesion and underwent radiotherapy. So in our study, we used 162 of our full cohort to determine allele frequencies in the population. We used 143 patients in our early toxicity analysis, and 125 patients reached or obtained the criteria to be part of the late skin fibrosis analysis that we did. We then graded toxicities using the common criteria for adverse events and a standardized toxicity protocol, toxicity assessment protocol. And we looked at early toxicities, which included mucositis, xerostomia, as well as dysphagia. And in late toxicities, we looked at skin fibrosis. So if the toxicity was graded 0 to 1, it was considered a control, and 2 to 4, it was considered a toxicity. And the toxicity was scored from clinical records, meaning that the study was done retrospectively. We then extracted DNA from blood samples that we obtained from the patient, and we did genotyping using mass array from Incapa Biotech. And we designed a genetic panel using SNPs from the literature review as well as bioinformatic databases. We then did a statistical analysis as well using SPS version 26. So our results, we found that about 50% or 51% of the patients experienced mucositis as an early toxicity. About 28% experienced xerostomia as a toxicity, and about 30% experienced dysphagia as a toxicity. And for skin fibrosis, about 25% experienced late skin fibrosis as a toxicity. Our cohort included a colored population, about 72%. We had Black African populations that we can find in South Africa. They were about 17% of the study. There was 9.1% Europeans included in the study, and one African patient as well, but from outside of South Africa. So these are our results. So for mucositis, we found that ERCC5, the SNP specifically RS17655, showed an increased risk for developing mucositis. So patients who had the SNP had an almost double increase in their risk to develop it in both the heterozygous, additive, as well as dominant models. And in XRCC1 or S3547, the SNP showed a protective effect in the development of mucositis. And then we had CHEK1, which plays a role in cell cycle. So RS11220184 also increased the risk of patients experiencing mucositis. And then we also compared how frequent these SNPs are in our study population, which is the South African population, compared to reference populations that we found on sample. So we can see that these SNPs were quite frequent in our study population and was quite frequent, equally frequent in the African population as well. And then when we looked at xerostomia, we found that ERCC1 as well as XRCC1, which both play a role in DNA repair, RS111615 had a protective effect against the development of xerostomia, whereas RS1799782 also had a protective effect against xerostomia in all of these models. And we have BRCA2 RS1801406, which increased the risk for a patient developing xerostomia. And if we look at the frequencies of these significant SNPs, we can see that RS11615 was quite frequent in our study population, but in general was not frequent in African populations. And we have RS1801406 and RS1799782 also present and frequent in our population. So when we looked at dysphagia, we found that TANC1 RS6432512 increased the risk for the development of dysphagia, and it increases almost 6 times in the homozygous model. So patients who had 6 times the risk for developing dysphagia if they had the SNP, as well as RS264588 also had a almost 6 times increase in the homozygous model to develop dysphagia. And this is actually quite interesting because TANC1 plays a role in the repair of muscles. So it brings about the question, is it because the radiation could be damaging the muscles around the neck, etc.? And so we have ERCC1, which plays a role in DNA repair. So RS11615 had a protective effect against dysphagia, and TFG1 beta 1, which plays a role in fibrosis and inflammation. RS480345 increased the risk for dysphagia, whereas RS2279744 had a more protective effect against dysphagia. And we can see these are the frequencies in the population. So we can see that RS6432512, which is the TANC1 SNPs, and RS264588, both had very high frequencies in our population as compared to other populations. So that is quite interesting. And then we see that RS1165, again, it was less frequent in African populations, but quite frequent in our study populations. And again, with RS227, 9744, about the same trend. And then RS4803-455 was quite frequent in both the SA study population as well as the African population in general that we would find on sample. So then we looked at late skin fibrosis, and we found that transforming growth factor beta 1, RS1 8000469 increased the risk for patients to experience late skin fibrosis, as well as RS224174 also increased the risk for patients to experience late skin fibrosis. And XRCC1, which plays a role in DNA repair, RS3547 also increased the risk for patients experiencing late skin fibrosis, and TP53, RS104-2522 also increased the risk for patients experiencing fibrosis. And then we had CCNU1. It had a more protective effect against late skin fibrosis, as well as ERCC1 had a protective effect as well. Okay. And if we looked at how frequent they were in our population, they were very well present and very well frequent in our population. Okay, so we can see that as well in figure 4, 5, and as well as figure 6. Okay, so discussion and conclusion. To our knowledge, this is the first radiogenomic study done in South Africa, and we have discovered potential genetic biomarkers to predict the onset of early and late toxicities following radiotherapy treatment in South Africa. And some of the studies like TANSI1 provided novel insights into genetic variants associated with radiotherapy-induced toxicities. The significant markers in our study have not been investigated or have not been significantly associated with radiotherapy-induced head and neck toxicity in other studies. So by identifying SNPs that are both relevant to the radiation response and prevalent in African populations, we could develop targeted genetic panels that exclude unnecessary variants and thereby making genetic testing more effective that way. And by investigating these genetic associations in previously understudied populations, this research aims to contribute to the development of more personalized approaches to radiation therapy in head and neck cancer treatment. It also is hoping to contribute to inclusivity and eventually leading to improved toxicity prediction and better patient outcomes. So with radiogenomic studies like these, it will be possible to implement locally appropriate strategies to treat cancer. There is several limitations that should be considered of the study. So the study is constrained by the heterogeneous nature of the data, relatively small sample sizes, and instances of missing information. And additionally, there is complex interaction between genetic and clinical factors to determine radiation toxicity. And so therefore, these should require careful interpretation of findings, especially when you want to implement it in clinical settings. So in future studies, future research direction should include validation in studies with larger, more homogeneous patient cohorts, investigating gene-to-gene interactions in radiation response, and prospective studies to evaluate the clinical utility of these genetic markers, not only in South African populations, but also other African populations for better toxicity prediction. So our work provides foundational data for historically understudied groups and highlights the urgent need for inclusivity and large-scale ancestry-informed genetic studies in Africa. Okay, and future studies should validate findings in independent, ethnically distinct African cohorts. These efforts will improve the applicability of radiogenomic biomarkers and support the development of tailored, population-specific radiotherapy approaches across the continent. Developing countries represent more than 50% of the world's landmass and is home to more than 50% of people of the world. And so if we improve cancer outcomes in these countries, we will significantly improve it globally as well. So I'd just like to acknowledge the following people as well as DARD and the SAMRC for funding. And thank you so much, everyone.
Well done, Dr. Cairncross. That was amazing. And I want to commend you on your very important and impactful work that you are doing.
Thank you so much.
Just see if everything's working. Sorry, I did get disconnected for a time when I know you were asking a question and I couldn't answer. Sorry about that. Are there any questions from the audience? You're welcome to pop it in the chat.
Should I stop sharing my screen?
You can, but now I see my thing is just loading as well, so I'm not sure what is happening. So just if I get disconnected, everyone's welcome to ask questions. Samantha, I saw you mention that we need to use more homogeneous cohorts or that's what do you mean by that?
I just meant that when I did my study, I kind of had to group everyone together because of sample size. But I think if we use more ethnically homogeneous cohorts, then it would be better to make ancestry stratification when it comes to the results.
Okay, I see. Is there no questions from, so we have a project going on in our department as well, the adult squamous cell carcinoma project. So that's Dr. Dada Timilola is the PI on that. So I'm sure he has lots of questions he'd like to pick your brain on. But what were the main challenges you found when you were conducting this particular project?
Well, the main challenges would include that a lot of patients, we could not include in the study because, say, for example, there's incomplete clinical records and things like that. And, when patients sometimes experience, especially when it comes to late toxicity, because late toxicity can sometimes occur or have an onset like 10 years following radiotherapy or 20 years following radiotherapy. And those patients never come back thinking, oh, they had a late side effect or something like that. So it was those type of challenges that I faced when I did the study.
Very interesting, Samantha. I understand what you're saying. So my screen is just loading, so I don't know if there are any questions. So you can stop sharing, and then if you do see any, you can tell them to unmute. Sorry about this.
Okay.
Technical
It's
issue.
just my screen is frozen as well. Sorry.
Oh, yours also. Is
Yeah, so I can't see
frozen?
anything. Yeah, I can't
Mine
even
is also
exit.
frozen.
My hand is messed up. If you can't see my screen, I can
Okay,
see
go
you
ahead,
guys.
Dada. Sorry, my screen is also frozen, so I don't know what's going on today. You can go ahead.
Can I pose a question?
You're welcome to.
Hello?
Sure. Sorry,
Yeah,
I can't see who's asking.
great. You can hear me, right?
Yes, go ahead.
I can hear.
Yeah, great presentation, Dr. Samantha. I just think maybe this question ties in with what was previously asked about the cohort of your patients, and also what I observed on the stats screen with that confidence index that's so wide. So this is why I'm just posing a basic question. If you can just clarify if the patient population was restricted to a specific histology and how you see tumor histology interacting with these predictive toxicity markers.
So unfortunately, if I had to just use like a specific histology, the sample size would be very small. And in general, this is quite a small sample size. So, and the toxicity that we found when it came, I know which one you're talking about with the wide confidence interval. So that is just because of the small sample size. We're hoping to increase the sample size and then to see how that confidence interval change, as also how the p-values will change. So yeah, that I know which one you're referring to. I just can't move my screen or anything like that to go there.
No, it's fine. It's just because different histologies like squamous cell would have a different dose as your sarcomas of head and neck cancers, because head and neck cancer is very general. But different histologies would get different doses and the outcome might be different.
Yes, so we did correction for any type of the accumulated dose. We did corrections. We looked at whether the histology would be a significant covariant. So that is how we try to overcome that. But in general,
Thank you, Dr. Samantha. That answers, yeah. Thank you so much. Great presentation, I must say. Fascinating.
Thank you.
Good afternoon, Dr. Samantha. Can you hear me now?
Good afternoon. I can hear you.
Yeah, I was talking before. I didn't know I muted myself from this end. So I thank you for the presentation and thank you for the good work that you put together. And I just have a few questions for you. First, I know during your presentation you talked about different groups in your cohort. I mean, the colored, the black, and the European population. I want to ask, did you at any point did something like a secondary analysis to look at the effect on the different population group?
I did treat it as a covariate. So I did look to see if the ethnicity would significantly play a role in whether a patient experienced side effects or not. So I did do that. And when it came to dysphagia, I think that European population was independently associated with increased risk, but at the same time, the p-value was like 0.049, so it wasn't too significant. I don't know if that answers the question.
Somehow. But I was actually thinking possibly you look at the different ethnicity group in your cohort and look at the effect on the SNPs and see if it will differ, if rather than instead of you grouping the population together, having just one
I
group
understand,
of population.
Prof. Okay, I understand now, Prof. Yes, I didn't do it for my PhD study, but that's something I would still love to do to see whether or not they would be they would differ between those population groups, Prof.
Thank you, Samantha. Dada, do you have another question? Your hand is up. Are there any other questions I might have missed in the chat? Unfortunately, I'm having terrible technical issues today. Samantha, are you still there as well?
I am still here.
Okay.
My screen finally unfroze.
Wonderful. Okay. Mine disconnected and reconnected. That's why I don't know if
Can you
anything
hear
happened.
me?
Yes, Dada, go ahead.
Oh, what's happening with it's just getting, I'll be talking, it just flicks along the way. Seems you're having some technical issue today.
Gremlins in the system today, for sure.
Okay, so I was actually asking that in your group the group you use for control, I think you said you use the people with the 0 to 1 low toxicity group, if I'm correct. So,
Yes, Prof.
yeah, so I'm just thinking that is it possible that if you have what difference will you make if you have another control group that have not actually started taking radiotherapy, using them as control group? Can you speak to that?
Can you just repeat the question quickly, Prof?
So
Sorry about that.
the group you use for control, I believe they've started also, they've had radiotherapy already. You just use them as control because of the low toxicity, which is 0 to 1. So the question is this: if I'm actually asking this question based on one of your conclusion. So if you're using a group of head and neck cancer patients that have not commenced radiotherapy, would that make a difference from the findings in terms of your finding, rather than using the low toxicity group as control?
That's something I can look into, Prof, but I don't know if it will work for the study because we are looking for patients who have not experienced toxicity. So if they didn't undergo radiotherapy, then we don't know whether or not they would have experienced toxicity or not.
So, Dr. Dadia, are you referring to a prospective study design? Samantha, was your study prospective or retrospective?
It was retrospective. Oh, I see, a prospective.
Yes.
That would definitely be something to do in the future. A prospective study.
Thank you.
Thank you.
Samantha, is your group continuing with this study, moving forward, or what's next?
Yes, I'm also hoping to continue with the study to improve.
I think Ima
increase
is
the
having technical issue also.
I am, yes, but you can continue.
Yes, I think Prof. Monji Benjado is continuing with the study as well, and I also want to continue with my study. I just took a short break off the PhD to recover, but I'm hoping to continue to continue and make the sample size bigger. And I still have a lot of results that I did not include in my PhD, but I would love to include as publications. So yes, I'll definitely love to continue.
And are you considering doing additional sites if you do continue?
Yes, I'm looking to go to other countries in Africa as well and include them in the study. I've spoken to a few people, and I'm hoping that things will work out as I'm getting into research again.
Okay, well, hopefully someone in this group might reach out to you to continue the study in different countries.
That would be amazing.
We have quite a number here on this call. Are there any other questions? Anything I may have missed in the chat? Doesn't look like it. Well, I think you answered all the questions, Samantha, and I thank you very much for attending to it today. I do apologize for all the technical hitches. I don't know what's happening with today. And I wish you luck and success on your further studies.
Thank you so much, and I just like to thank you so much, Iman. And I would just like to thank the African Society for Dental and Facial Cranial Genetics for inviting me to do this webinar. And I hope that we do get to work together in future. I have a lot of projects that I would like to collaborate with you on. And so I hope that you invite me for that coffee you promised me. And
sure.
We can talk.
Okay. Everyone, if you want Samantha's email, you're welcome to message us on our society email and we'll respond to you. Otherwise, you can follow on LinkedIn. I think I did see you on there, and send a message through that. Okay. Thank you, everyone.
Thank you so much.
We'll see you next month. Goodbye.
Bye-bye.
Bye.
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 Bye.